Chapter 5 is all about the plasma membrane and the transport that happens across that membrane. Now that's probably something you've never thought about before, but bringing components or chemicals into and out of the cell is vital for cell function, whether that is an individual cell such as a bacteria or a cell that's part of a larger organism. For example, we have two brain cells here, and this white structure that's drawn here is the plasma membrane. There's a lot going on here, but you might notice that there are proteins that are reaching out across that membrane, and those proteins are important in bringing little molecules in and out of the cell. Now those molecules can move through some proteins, or they can be enclosed in a plasma membrane and brought in sort of in a package. So I've labeled this as endorphins. I don't know if these are endorphins or not. I actually have no idea what the artist had in mind here. But endorphins are hormones, and hormones are an example of a molecule that might be moved into and out of a cell. So the goals for Chapter 5 are to recognize the structure, components, and characteristics of the plasma membrane. So I might ask you to construct a diagram of that plasma membrane, or I might ask you to label a diagram. So you're going to want to be familiar with all of the major structures and key components of the cell membrane. You'll also need to be aware of the characteristics of that membrane. And by this I mean physical properties. Next, you'll also need to compare and contrast the mechanisms that move particles and molecules across the plasma membrane. And we've got quite the list here. We'll go through each one of these. And these include simple diffusion, facilitated diffusion, active transport, membrane-mediated transport, and osmosis. So let's take a look at the plasma membrane. The plasma membrane is really important because it separates the cell from its surroundings. In other words, it acts as a boundary for that cell, and it controls what moves in and out. And it does this because it is selectively permeable. Biological membranes have selective permeability, which means that it allows some substances to cross freely and blocks others, or just makes it more difficult for some to cross. So some molecules can just move right through that plasma membrane, straight across. Others have to have essentially a gateway or a passageway through that cell membrane. And the way that it does this is based on its chemical properties. Now that might be the properties of the molecule itself that is moving, or it might be properties of the plasma membrane. Usually it's a combination of both. So we have a simple diagram down here on the bottom, and it depicts just some of the types of molecules that you might find working to transport molecules or substances across in order to aid in the permeability of the membrane. So we've got a channel protein here. Molecules might move through this channel. They might move freely, or they might be blocked or controlled. Molecules can also move just straight through. Some molecules can move just right through that plasma membrane. Over here on the right, we have an aquaporin. An aquaporin is a special molecule. Aquaporin is a special molecule a special protein that allows water to move freely. And water molecules, which are indicated in the red and gray here, will move right through that protein across to the other side. So it's basically making a channel for that water to move through into or out of the cell. The ability of a cell to be selectively permeable comes from a characteristic of phospholipids. Now, the main components of a plasma membrane are phospholipids. And if you recall from chapter 3, we learned that phospholipids are amphipathic. Amphipathic means that there are some regions that are hydrophobic and some regions that are hydrophilic. Now, those phospholipids have a phosphate head and they have a fatty acid tail. And if you remember from chapter 3, we learned that that phosphate head is very hydrophilic. Remember, that means water-loving. And those tails are very hydrophobic because they are lipids. Now, you might remember from chapter 3 that phospholipids will self-assemble into a double membrane. So phospholipids create a bilayer, 2 layers of phospholipids. One where the heads are attracted to water inside or outside the cell. And then the tails are repelled by that water. And so they orient in a double layer on the inside of those phosphate heads. Now, this amphipathic nature is really important because in general, it's going to block hydrophilic molecules. So hydrophilic molecules aren't going to be able to move freely. So molecules that have charges like ions or just large polar molecules aren't going to be able to move freely because that center layer that is hydrophobic is going to block them. So the water molecules and the hydrophilic molecules are going to interact at the edge with the water, but they're not going to be able to pass through the hydrophobic layer. And that's going to contribute to that selective permeability. Because of this amphipathic nature of those hydrophilic and hydrophobic phospholipids, we end up with an organization structure that allows for certain molecules that are hydrophobic along the center to become embedded in that plasma membrane. And then anything that's hydrophilic might be attached to the outside. We can also embed things right inside that hydrophobic layer. And what we end up with is something called the fluid mosaic model for cell membranes. And we call it this because membranes are, in air quotes, mosaic, meaning a collection of pieces or a collection of parts of proteins in a fluid bilayer of phospholipids. So we have that bilayer made up of our phospholipids, and we have proteins and other molecules that are embedded in that bilayer. Those proteins can be transmembrane, meaning they stretch all the way across the membrane. They might be peripheral, which means they're stuck to the outside or to the inside, but not embedded in the layer. And those transmembrane proteins can act as transport proteins. Now we can also have integral proteins, which means they're embedded, but they're not going all the way across. Our focus is going to be on those transmembrane proteins, because those transmembrane proteins are the ones that are going to be transporting molecules across and that contribute to that permeability of the membrane. Now you probably noticed there's a lot of other things going on this structure here. We've also got the extracellular matrix. Remember from chapter 4 that that is a structure that helps provide support on the the outside of the cell. Inside we get support from the cytoskeleton. And that extracellular matrix is of course actually attached to the cell, and it is attached by glycoproteins and glycolipids. Glyco meaning carbohydrate, so they are carbohydrate proteins, and they are carbohydrate lipids. They're molecules that are two-part molecules. And those glycoproteins and glycolipids both work in attachment to the extracellular matrix, and they might also work in communication within a cell, basically being a signpost or a flag to other cells in the body or in the organism for what they are and what they do. They might also be involved with transferring messages between cells. And then we also have embedded in our plasma membrane cholesterol, and we're going to come back to that. But cholesterol helps with the fluidity of the membrane. So part of the name fluid mosaic model is fluid. That means that the membranes are not locked in place, and the components can actually move because they are not locked in place either. Instead, they are held together by hydrophobic interactions. And anything that is embedded in a membrane is going to have a hydrophobic region that is attracted to those phosphate tails, and a hydrophilic region that is attracted to the water and the hydrophilic heads. And that's going to help hold them in place. They're basically held together by attractions and repulsion. Now, if we're saying they're not locked in place and they're fluid, that means that they can move. And those parts are going to move laterally. So these pieces are not going to pop in and out, but they can shift side to side and front to back. And so a protein that is embedded here could move around the other parts, could move back, could move forward. There are a couple of factors that can cause changes in the membrane fluidity or to help regulate how fluid that membrane is. One is the type of hydrocarbons in those fatty acid tails, and another is cholesterol. So if we have more unsaturated hydrocarbons, remember an unsaturated hydrocarbon is one that has a kink in the tail, those membranes are going to be more fluid. And the reason that they are more fluid is that those unsaturated tails prevent packing or close packing of those phospholipids. So organisms that live in areas that are colder are probably going to have membranes that have more unsaturated hydrocarbons so that those membranes don't become as solid. We can also have less fluid membranes. So if you have more saturated hydrocarbons or hydrocarbons that don't have kinks in the tail, they're going to pack together more tightly. So saturated tails are packed more tightly. So there is less movement. So cells that are going to be in warmer situations or hotter situations are going to have more saturated hydrocarbons because you don't want your cell membrane to essentially melt and fall apart. The cholesterol can also affect membrane fluidity, and it does so by interfering with that phospholipid movement. So we've got two things going on here that are going to affect how solid and how fluid that membrane is. One are the makeup of the hydrocarbon tails. The other is the presence of cholesterol. So cholesterol can do two things at either end of the temperature range. As temperatures decrease, more cholesterol in a membrane decreases packing and increases movement. In other words, it essentially keeps the membrane from freezing or becoming too solid because it is blocking that tight packing. When there's a temperature increase, cholesterol reduces fluidity by blocking movement of those phospholipids. So it keeps those phospholipids from moving around. So the same molecule in different situations is going to both reduce fluidity and it's also going to hinder solidification. Now, the amount of cholesterol in a membrane is probably not going to change much across a single organism, but different organisms might have different cholesterol components in their membranes because of the environmental situations that they are in most frequently. So selective permeability means that only some molecules can cross freely. And lipids and proteins function together to help regulate that transport. Lipids can block molecules from moving directly across that plasma membrane, but proteins can allow certain molecules to move freely. Now, cells exchange several different types of chemicals with their environment, many of which are food type molecules. So sugars, amino acids from proteins, nutrients, waste, gases such as carbon dioxide and oxygen, ions, and of course water, which is really important for the cell. So we're going to talk about the ways in which these molecules move. And most of our transport mechanisms that we are going to focus on are moving single molecules, one molecule at a time, although we can also have bulk transport of larger molecules or particles. So aside from this vague idea of particles moving across a membrane, why do we care about the plasma membrane? Well, there are a few diseases that affect the cell membrane, both of which you've probably heard of. You might even be very familiar with some of these. One is Alzheimer's disease, and Alzheimer's is caused by a plaque formation along the cell membrane. and this inhibits ion movement. Now, there's an additional product of this plaque that causes twisted molecular fibers inside the cell. So it's not just the cell membrane, but that's what we're focusing on here. And when it inhibits those ions, it also disrupts cell-to-cell signaling. Now, you probably know that Alzheimer's can be a really nasty disease, and the reason is that it leads to cell death. Specifically, neurons. And this can lead also, or because of that cell death, it can lead to memory loss, difficulty with daily tasks, problems with language, disorientation. Another disease that you've probably heard of is cystic fibrosis. In cystic fibrosis, chlorine ion transporters don't work. And we'll talk about what that means, what a chlorine ion transporter is, in a few minutes. But basically, it's an ion transporter that needs to regulate the movement of chlorine across the cell. When these ion transporters are faulty, they aren't embedded correctly in the plasma membrane, and then these ions aren't transported. This causes a buildup of fluid and mucus along membranes and organs. So this also hinders movements of other molecules and causes those organs not to function correctly, particularly the lungs, which can cause issues with breathing. Now, there are other effects of cystic fibrosis as well, other effects to organs, but they all come from this lack of chlorine ion transporters and this buildup of mucus across the membrane. So we're going to talk about some specific molecules and how those molecules move across membranes. I'd like you to pause, take out a sheet of paper, and try to answer these questions based on the information that I've given you about the physical characteristics of a plasma membrane. So hydrophobic molecules dissolve in the membrane and pass easily without the help of any sort of transport molecule. Why is that? Polar and ionic molecules, including water, don't pass easily or quickly. Now, water molecules can pass through the membrane, but in general, they don't pass through the membrane at a high enough rate, and we require aquaporins. So why is that? Why do polar molecules and ionic molecules get stuck on the outside or inside and can't move freely?